lately too. Welcome everybody. We'll wait a few minutes to let everyone join, and then we're going to talk about duration of antibodies today. And Arnold Catt has offered to summarize that New England Journal paper as well. So we'll have a total of
got Josh's comments, and apparently the conclusion that I reached is the same as Florian reached.
That it doesn't really look like rapid decline.
Yeah, rapid decline. It's not linear.
Yeah.
Decline is not linear.
I think
but you can get a couple of papers out of it in the process.
Yeah, that's true.
Or letters. Letters have become the new norm.
Yeah, and speaking of that, I think we're actually gonna start with a letter, which is looking at some of the data from the original SARS as a theoretical basis for decline in antibody titer for SARS-CoV-2. And I don't know if Samantha is on yet, but she's going to be oh, it looks like I do see her there.
Does anybody know, to develop some conspiracy theories that I was just thinking about, whether anyone was doing any work in Wuhan on live SARS vaccines? if we want to really get the pot boiling, I had some information that the whole H1N1 pseudo pandemic was based on work being done on live vaccines, experimental work being done on live vaccines in China.
Wasn't one of the H2N2 outbreak? Wasn't the H2N2 outbreak?
Yeah, well, you
supposedly a release from a Russian lab.
Yeah, we have that too, but becoming widespread, I think requires a lot of seeding in the population.
Yeah, that's probably right.
The more we see of this virus, I'm thinking that it's very unlike the original SARS in the way it's behaving, unfortunately.
How did the original SARS behave?
The transmission patterns were somewhat similar, but it sure didn't spread it this easily. It was contained completely by non-pharmaceutical interventions and then went away.
Do you think part of that is that the full extent of the original SARS is not really well known, since they weren't looking for mild, asymptomatic, it wasn't PCR?
That's true in terms of the spread, because I'm sure there were multiple introductions into the US. But what that really means is that there were more defective viruses or whatever.
Sure.
That didn't allow the widespread dissemination. The problem here, and this was pretty obvious a month or two into the whole thing, is that it's everywhere.
Yeah, and the original SARS the transmission seemed to be driven even more so by super spreading events than this, right? So maybe that's part of it.
Well, it was spread by super spreading events, and I reluctantly have to conclude that this one spreads a lot better, which I didn't want to conclude early.
Yeah.
Just because I didn't like the
implications of that.
Consequences of drawing that conclusion. But it sure is behaving very strangely. We've rarely had a virus that didn't show any kind of summer seasonality or respiratory virus, just in general.
Although I will say that the
It could be worse.
it could be worse. The H1, I mean, H1N1 pandemic in 2009 started out in the spring and summer. I mean,
But it went away in the summer.
It did.
Out in the summer, and then
peaked again in the fall.
The schools up. But it was driven by young children. That was the other thing.
Right.
But this one is there may really be seasonality based on what's going on in Australia and places like that, which are in. Remember, it's July, which is equivalent to January,
Mm-hmm.
and they're beginning to see real upticks. So it may be that in temperate areas you see more of it, but who knows? You can come up with any theory and write a paper. Or a letter.
That's right. All right. Well, let's go ahead and get started with today's topic, which is antibody duration, and we're going to start with Samantha. If you want to share your screen.
Okay, yeah, I can share right here.
Yep.
Okay, so hopefully you guys can all see it. So I'm Samantha Dell'Imperio. I'm a rising sophomore at Syracuse University, where I study biology and medical anthropology. And the paper I'll be discussing in my presentation is the duration of serum neutralizing antibodies for SARS-CoV-2, lessons from SARS-CoV infection, from the Journal of Microbiology, Immunology, and Infection. So this paper draws upon two important concepts that I just linked, and they are antibodies and COVID-19 and the epidemiology of SARS-CoV. So the basis of this paper, or this letter, I guess you can say, is that a study showed that SARS neutralizing antibodies can block SARS-CoV-2 entry into target cells in vitro, suggesting the two viruses have a cross-protective epitope and have similar immune protection against reinfection. So by the way, this paper is quite short, so I took figures and statistics from its references. But an important note about the topic is that after viral infection, B cells produce these protection-specific antibodies like immunoglobulin G and neutralizing antibodies to block the virus from entering host cells and to prevent against reinfection. So several studies have shown that these two antibodies the paper discusses, immunoglobulin and neutralizing antibodies, are highly correlated. And so there are several studies here that display that. So there are varying lengths, and the first one is a 2-year cohort study of both antibody levels in SARS-CoV patients. So it takes 56 cases from a hospital in Beijing, and at 4 months, the immunoglobulin and neutralizing antibody levels peak. After 16 months, the titers gradually decline. Before 2 years, both antibodies were detectable in all cases, but then after that 24-month mark, 11.8% of the samples turned negative. And these researchers published another paper where they did an extended follow-up. So at 36 months, immunoglobulin antibodies were detectable in 74.2% of patients, and neutralizing antibodies in 83.9% of patients. So not 100%, but still high percentages, and these antibodies, they have similar percentages as well, which suggests that they are highly correlated. So the next study is a 3-year study of just immunoglobulin antibodies in SARS-CoV patients, and it takes a larger sample of 176 cases from 7 designated SARS hospitals in Shanxi, China. So at day 7 after symptom onset, 11.8% of the serum samples are immunoglobulin antibody positive. Then from day 90 to 200, all the samples are immunoglobulin positive. After 1 year, that decreases a bit to 93.9% of samples. After 2 years, it goes to 89.6%. And then at 3 years, only 50% of the convalescent patients had SARS-CoV specific immunoglobulin antibodies. And these researchers also did a selective cohort study from the original 176. So they just took 18 patients from this group, and they did a separate analysis of them at 6 months and then 1, 2, and 3 years to obtain an immunoglobulin trend that they thought would more accurately represent the convalescent SARS patients. So at 6 months and 1 year, all 18 of these patients had positive immunoglobulin, and then at 2 years, only 1 became negative, but then at 3 years, only 55.56% are immunoglobulin positive. So this is quite similar to the previous study in which at 2 years, the percentages of the antibodies were pretty high, but then at 3 years, it dropped to only about half of the sample. And then the longest longitudinal study was a 6-year study of just, again, immunoglobulin antibodies in the SARS-CoV patients. And they found that the specific immunoglobulin antibodies were undetectable in 21 out of 23, or 91.3% of the recovered patients. So if you follow the black line here, you can see that decline, and the numbers above each black line are the number of serum samples detected at each visit. And you can see this goes up to 70 months, so a little bit after that would be the 6-year mark. And finally, there's a figure from the original paper that sort of gives a nice picture of the 3 studies I just mentioned. And it starts with day 7 with only 11.8% of the immunoglobulin antibodies being present in the patients. But then from 3 to 16 months, it's 100%, then starts to decline. And then you see around 3 or 4 years, it's about 50% of the patients have it. And then the longest study, 6 years, only 8.7% of patients have the immunoglobulin antibodies. So taken together, these studies show that the immune responses to specific antibodies were maintained in over 90% of the recovered SARS-CoV patients for 2 years, but then declined a bit in 3 years and was very low in 6 years. So going back to the main idea and why this is important to study SARS-CoV as a basis for SARS-CoV-2, there are implications of similar immune protection between these 2 viruses. So although the specific immunoglobulin antibodies to SARS-CoV significantly declined in many recovered patients from the 2003 epidemic, these antibodies probably wouldn't be sufficient in protecting the individuals from SARS-CoV-2 infection. There's still evidence that SARS-CoV vaccines and neutralizing antibodies can serve in treatment for COVID-19. And for this reason, it's crucial to detect the antibody titers for convalescent COVID-19 patients today. So that's my summary. Does anyone have any questions or comments?
So one of the papers was from Beijing. Where was the other one from?
1 second, let me just go back to it. So, yeah, so one was from Beijing. Actually, 2 of them were from Beijing, and then the 3rd was from Shanxi, China.
Okay.
Yep.
I don't know that we've seen any papers or studies, epidemiologic studies in those cities in Asia that were hit by SARS in terms of COVID.
Mm-hmm.
But I'm sure they're looking.
Yeah.
And surprised we haven't seen anything out of Hong Kong yet.
Yeah, that's what I was just going to say.
Major outbreak.
Mm-hmm.
Seems like Hong Kong is the spot.
Yeah.
All right. Well, if there's no questions for Samantha, I think Yonita was the next presentation. And now we're going to move on to some actual SARS-CoV-2 data.
Yeah, hi, my name is Yonida Traco. I'm a rising junior majoring in pharmaceutical sciences at the University of Michigan. I'm sharing, let me share my screen right now. If you can't see my screen or you can't hear me well, please let me know. The paper I summarized was a paper that basically looked at the neutralizing antibody response into SARS-CoV-2 in a COVID-19 recovered patient cohort and their implication. The virus specific neutralizing antibodies are very important in creating an immune protection against viral infections. They have been widely used even in the past as a passive antibody therapy for SARS-CoV and Ebola virus. They have also been used in evaluating the efficacy of vaccines against smallpox, polio, and influenza viruses. In this study, they sampled plasma from 175 adult COVID-19 patients that were admitted to Shanghai Public Health Clinical Center. These plasma levels were screened using a sensitive pseudotype lentiviral vector-based neutralization assay. And all the participants had mild to common symptoms with COVID, and they developed peak neutralizing antibodies levels 10 to 15 days after the onset of the disease. The study, as they analyzed the levels of the neutralizing antibodies in the different participants, showed a lot of variation, as I will show in a minute. The first thing that they studied is that the plasma levels from the COVID-19 patients, they contained highly SARS-CoV-2 RBD, S1, and S2 specific binding antibodies. These plasma levels, they tested them, and they were not effective in inhibiting SARS-CoV infection using the pseudo-lentiviral vector neutralization assay, but at the same time, the plasma showed some cross-binding activity with the SARS-CoV antibodies, which shows that, which could be explained by the conserved epitopes between the SARS-CoV-2 and SARS-CoV spikes proteins. Actually, in the study, they said that these spike proteins, they have like a 77.2% identical amino acid sequences. So that could explain why there was some cross-binding activity, but not inhibition. And also they said that this experiment, this data suggests that the immunogenicity between SARS-CoV-2 and SARS-CoV is very different. At the same time, they looked at the levels of the neutralizing antibodies in these participants, and they observed very low titers of SARS-CoV-2 neutralizing antibody in about 30% of the patients, which had they measured it with the ID50, which was for the 30% was less than 500. The values for ID50 go from below 40, which is barely detectable, to up to 2900, which is very high. Interestingly enough, 10 patients presented an ID50 of less than 40, which is even below the detection levels. 70% had showed a medium to low neutralizing antibody titers, 39% a medium to high, and 40% a very high neutralizing antibody titers. These plasma levels of the neutralizing antibodies were measured again for these participants 2 weeks after their discharge to see if there was any difference. And in fact, there was no difference from the moment that from the initial point that these neutralizing antibodies were measured. At the same time, it's interesting to point out that these 10 patients, or even the 30% patients that have very low titers of neutralizing antibodies, they were still recovered, were able to recover without developing very low neutralizing antibodies. And as the study pointed out, it's interesting to analyze further how this group of patients were able to recover without the help of the neutralizing antibodies. And at the same time, the high levels of SARS-CoV-2 specific and neutralizing antibodies in elderly and middle-aged recovered patients, there was this positive correlation, obviously, between age and neutralizing antibody titers, which can explain why elderly have a very bad outcome with this infection. However, elderly patients' plasma levels show significantly higher levels of spike binding protein antibodies targeting S1 and S2 compared to the young participants. And at the time of the admission, elderly patients had higher blood CRP levels, which is a marker of inflammation, and lower lymphocytes counts, which might suggest that the high neutralizing antibodies levels in this group of patients can be due to a stronger induced innate immune response. However, it's very interesting to notice that there was no difference between the patients' ages and the time they spent recovering in the hospital. As this graph shows, I hope it's enlarged now. It shows that the age groups from 40 to 85, they have an ID50 that is above 1,000, which is quite high for the neutralizing antibody titers. And the study left a lot of question marks, so things that they suggested that could go further investigated. So I'd like to present even the limitations that they also included in the study that could be confronted with the data and compare. So first of all, the viral RNA was not detected in these patients' blood. There was not enough information available about the kinetics of the viral load, and severe COVID-19 patients were excluded from this study because passive antibody therapy was administered before the sample collection. So all these patients that participated, even though it's a small group, they all have mild to common symptoms. They were not ICU patients or patients with severe symptoms. And that is all. If you have any questions or comments, please let me know.
Okay, great job, Judita. And then I think Ashley was going to be next.
Yep, I will share my screen. Okay. So my study was antibody dynamics to SARS-CoV-2 in asymptomatic and mild COVID-19 patients. This study evaluated antibody dynamics among 177 asymptomatic and mild individuals in Wuhan, China, from February 17th to April 28th. All right, so the authors screened 177 individuals using nucleic acid testing, which is acronym NAT, IgM and IgG antibodies from commercial serum kits were evaluated against the SARS-CoV-2 nucleoprotein N and spike S proteins. Individuals were grouped into classifications based by COVID-19 severity, NAT and IgM, IgG response for a study sample of 177 participants with 63 healthy contacts of COVID-19 cases, 63 asymptomatic cases, and 51 mild cases. And the authors also evaluated neutralizing antibody responses. So this is the table 1 from the paper. I can zoom in a little bit. But just to give an idea, as I said, they were categorized based on the antibodies. So we have healthy contacts over on the left, and then asymptomatic had 4 of those categories, broken down by NAT and the antibodies, and then mild had 5. So just to give an idea of what antibodies were dominating in their different fields. They saw different ones for mild and then also different ones for asymptomatic, which is going to come into play in their results. Okay, so the S1 and N IgM, IgG responses showed in asymptomatic individuals 7 days post-exposure and were greatest after 17 to 25 days post-exposure. The S1 IgM and IgG responses and titers of neutralizing antibodies disappeared after 2 months post-exposure. And whereas the S1 IgM antibody responses showed in mild individuals after 1 day of that symptom onset, and they increased until 29 days after onset. Mild patients experienced greater N-specific responses for IgM, IgG, and remaining for over 65 days. 63.5% of asymptomatic cases and 19% of healthy individuals induced titers of neutralizing antibodies, with a mean NT50 of 1 to 24 and 1 to 13 respectively. The neutralizing antibody response among asymptomatic individuals occurred after 7 days post-exposure and peaked after 10 to 25 days, and 88.2% of mild cases induced titer with a mean NT50 ratio of 1 to 269, occurring 1 day after symptom onset and peaked at 22 days. So then I have the 2 figures relating to this. So the first one is the dynamic change of S1 and N specific IgM and IgG antibody responses. And so let me zoom in. So the color coordination occurs up here in this legend, but the green represents healthy contacts, the orange is asymptomatic, and the blue is mild. So on the left, for both the top and the bottom for the 2 antibodies, we have days after exposure for the asymptomatic and healthy cases. And then on the right, we have the mild for days after symptom onset. So again, just kind of looking at the differences between the antibodies response here. And I'll get into their description of that in a second. But as you notice, the slope is going to be a little bit more extreme, a little bit more rapid on the left side of the 2 figures versus on the right for the mild. The lines indicate antibody responses against the virus at different times after exposure for the asymptomatic and healthy contacts, and again, after onset for the mild cases, with the bars indicating medians and IQRs. And so the other figure is similar, but it's looking at the titers and their dynamic change of neutralizing antibody in different groups. So on the bottom, let me see, I'll try to zoom in so you can see both. Up here, again, is just the figure legend, or not the figure legend, I'm sorry, the summary for the figure, and then on the bottom is going to be the figure itself. So in A, we'll see over here the comparison of titers of neutralizing antibodies in different groups. B is going to be the comparison of the titers of neutralizing antibodies in different subgroups of asymptomatic individuals and mild patients with that of healthy contacts. And then down here, C is the dynamic change of the titers of neutralizing antibody over time, with the lines indicating change of neutralizing ability at different times after exposure for asymptomatic cases in those healthy contacts, or after symptom onset for the mild cases. And then samples with neutralizing titers below that 1 to 10 were plotted at the dotted line with statistical significance in A, B, determined using an unpaired t-test, and horizontal bars indicating the mean and standard deviation. A little bit into their discussion. So overall, the authors found that the IgM and the IgG antibodies couldn't differentiate between asymptomatic or mild individuals, which they contributed to a potential infectiousness of asymptomatic individuals. And titers of neutralizing antibodies were low overall among those asymptomatic cases, and then they rapidly deteriorated following a peak of 10 to 25 days post-exposure, compared to that mild decrease, which peaked at 22 days and lasted for over 65. So that's the end of that paper. Are there any questions on that one? Okay. And then I will turn it over to whoever is next.
Thanks, Ashley. That was great. Amanda, I think you're going to wrap up our discussion today with the One Academy tool, and then Kat is also going to give us a brief summary of another paper that just came out recently this week.
Okay, thank you. Hi, my name is Amanda Sherwood. I'm a rising junior at the University of South Florida, and I'm currently studying cellular and molecular biology. So I'll just share my screen here. Okay, so before I zoom in and begin reading off of the notes, I just wanted to briefly talk about why I organized my notes this way. And I just wanted to present the main findings in the same order and in similar linearity to the paper. So the researchers approached studying antibody durations in 3 parts. So first they had to establish that PCR patients do produce antibodies, and that's going to be here. And then that antibody titers have been shown to correlate with neutralizing titers, which is going to be here. And then finally, they examined the longevity of these antibodies, and the conclusion is over here. So I'll zoom in. Okay. There we go. So, according to the authors, antibody function and duration in severe SARS-CoV-2 cases is relatively well characterized, but little is known about the duration and function in mild to moderate or even asymptomatic cases. And those constitute the majority of infections. Therefore, the individuals that take part in the study are the vast majority mild to moderate COVID-19 cases. So here, this is the seroprevalence of anti-SARS-CoV-2 spike proteins in the New York metropolitan area. So as of July 2nd, over 51,000 individuals were screened by the Mount Sinai Health System to recruit volunteers for a convalescent plasma therapy. The individuals screened either had confirmed SARS-CoV-2 infections by PCR or suspected disease, which is defined as a physician attributing suspected symptoms to SARS-CoV-2 or exposure to someone infected with SARS-CoV-2. So in order to determine the parameters of a viable candidate for plasma therapy, an enzyme-linked immunosorbent assay, or ELISA, was administered to the participants, and those with titers greater than or equal to 1 to 80 were considered to have undergone seroconversion. And another thing to note, that out of all the individuals that were screened, less than 5% of cases required hospitalization. So once again, the vast majority of individuals screened either had mild to moderate cases. And then here's just a side note, talking about the two proteins that have been used as target antigens for serological assays. So the nucleoprotein and the spike protein. And in this study, the spike protein is the target, and the researchers believed that it's potentially the only target for neutralizing antibodies, in contrast to nucleoproteins, where it's kind of unlikely that nucleoprotein antibodies would directly neutralize SARS-CoV-2. So then here are the results. So over 19,000 individuals, so that's 19,763, they screened as positive, and 32,063 individuals screened as negative. And of the positive individuals, 505 had a titer of 1 to 80, 943 1 to 160, 4,391, 1 to 320, 6272 of 1 to 960, and 7641 of 1 to 2880. So titers of 1 to 80 and 1 to 160 were categorized as low, 1 to 320 is moderate, and 1 to 960 and anything greater than or equal to 1 to 2880 is high. So from these results, the researchers deduced that the majority of the positive individuals have moderate to high titers of anti-spike antibodies. And apologies for the blurriness of the graphic. This is a preprint. This was printed in medRxiv, so it's not officially out on the press yet. So this is just a closer look. Figure A is just a pie chart breaking down the distribution of varying titers among positive individuals. And then here is B is showing the absolute numbers and the percent of individuals with titers of 320 over time. So around here, the program shifted from just plasma donors to employee screening. So anyone as an employee within the healthcare system of Mount Sinai. And they have a likely higher number of asymptomatic infections, which kind of the researchers assumed that that would probably cause the decrease in fluctuation in percentage above 1 to 320 and at later time points. And then once they determine that the majority of positive individuals produce antibodies, then it's determining the functionality of the antibody response. What good is producing antibodies if they don't exert neutralizing effects? So determining these effects can give researchers insight into understanding the possible protective effects of the innate immune system, the innate immune response, and to determine if spike antibodies exert any neutralizing activity. A quantitative microneutralization assay based on authentic SARS-CoV-2 was performed with 120 ELISA samples with known titers ranging from negative to 1 to 2880. Okay. And then here are the results. So from performing the assay, neutralization titers significantly correlated with spike binding titers. And although there is some variability, sera with 1 to 320 ELISA titers had a geometric mean, 50% an ID50 of approximately 130. And then for the 1 to 960 titer, it was approximately 1 to 75. And for the 1 to 2880 titer, it was approximately 1 to 550. So when considering any activity above background noise, approximately 50% of the sera within the 180 to 1160 titer range had neutralizing activity. 90% in the 1 to 320 range had neutralizing activity in all sera in the 190 to 1960 to the 1 to 2880 range had neutralizing activity. So these results, while they're not conclusive, they validate use of antibody titers to refer for plasma donation and further investigation into neutralizing effects at varying titer levels. So right here, figure A is a correlation analysis. and it shows a correlation analysis between ELISA titers on the x-axis and neutralization titers in a micronucleization assay on the y-axis. So the Spearman number, or Spearman R, is 0.87, which is pretty good correlation. And B shows the proportion of sera that exert any neutralizing activity in each of the ELISA titer categories. So we see the higher the titer, the more percentage of neutralizing activity. So after examining the correlation between seropositivity and antibody levels and the neutralizing effects of spike antibodies, now the researchers were finally able to assess the longevity of spike antibodies. So what they did was they evaluated the medium range stability of spike antibodies, and they recalled 121 plasma donors at various titer levels that had initially been screened at approximately day 30 post-symptom onset. And then here are the results. So these results were in agreement with earlier observations that seroconversion in mild COVID-19 cases might take longer time to mount. So this showed an increase in individuals who initially had an initial titer of 1 to 320, 1 to 160, or 1 to 80. So the mean interval between the initial titer measurement and the second was 52 days, setting the second time point at a mean of 82 days post-symptom onset. There is a slight drop from a geometric mean titer, or GMT, of 670 to a GMT of 642 when comparing overall titers. And the higher titer range of 1 to 2880 and 1 to 960 also observed a slight drop in titer between the two time points. So there is an exception in this particular study. There was one person that dropped from a 1 to 80 titer to negative, and the researchers potentially attributed this to the antibody serum titer being produced by plasmablasts, and those antibodies peak 2 to 3 weeks post-symptom onset, whereas the sustained or increasing antibody titers observed are more likely produced by the long-lived plasma cells in the bone marrow instead of the more short-lived plasmablasts. And then before we get to the conclusion, the researchers also noted that there was a similar report also put out roughly at the same time that produced results that found waning titers 8 weeks post-viral infection. So the results contrasted a little bit. So in asymptomatic cases, antibody responses disappeared after 8 weeks in 40% of the individuals, but the antibodies measured in the report were nucleoprotein antibodies plus a single linear spike epitope, which is why I decided to explain the difference between spike proteins and nucleoproteins. The report also shows relatively stable or slightly declining neutralizing antibody titers, which are in agreement with the study assessing the longevity. So this led researchers to potentially conclude that the stability of antibody response over time might also depend on the target antigen, which would once again open up avenue for more study. So then here's the conclusion. So whether infection with SARS-CoV-2 prevents reinfection, or rather for how long, it remains unclear. So what researchers have found is that with other common coronaviruses, neutralizing antibodies are induced and can last for years and provide protection from reinfection. Work done with non-human primate models infected with SARS-CoV-2, infection does protect from reinfection for some time. And then they've also transferred serum from infected to naive hamsters, which reduces virus replication significantly when the naive hamsters are presented with SARS-CoV-2. And vaccine-induced neutralizing antibody titers have been established as a correlate of protection in non-human primates. Spike antibody titers correlate with neutralization with authentic SARS-CoV-2 virus, and a significant number of individuals with antibody titers of 1 to 320 or higher show neutralizing activity in their serum. And consistent with data for other human coronaviruses, researchers found stable antibody titers over a period of 3+ months. So finally, establishing a correlate of protection along with continued study of antibody functionality, duration, and kinetics would greatly benefit vaccine development and assist in improving policies regarding the COVID-19 pandemic. As for any limitations, there weren't many stated explicitly. The main ones that were kind of touched upon were, one, that further investigation is needed on the neutralizing effects of antibodies, as well as neutralizing effects at various titer levels. And along with that, there are only some studies done with regards to whether or not antibodies protect from reinfection with regards to SARS-CoV-2. And all of them, at the very least in the paper, involve animal models. So once again, it's to be taken as, circumstantial evidence, but it's still quite promising. So that concludes the end of my presentation. Does anyone have any questions or comments?
When they talked about the common coronaviruses not having reinfection, what was their citation of that?
Okay, so I have the article pulled up currently.
Just curious, because we don't find that.
Yeah, so give me 1 second. They, obviously with papers, they tend to have a lot of citations.
A lot of conclusions.
A lot of conclusions. So I believe that's 23. Okay, so that's a paper done. It's by A.T. Huang et al. It's a systematic review of antibody-mediated immunity to coronaviruses.
Oh, okay.
Yeah.
It's a review.
Mm-hmm. And it's also it also says that it's been published.
Is that the one that was? Can you see where it's from? Because there's a group in Florida that was commissioned to do a review.
Mm-hmm. So this is also in the same journal in RxIV. So it's also,
okay. Right.
Yeah. So there's no DOI that I can go to.
It's unrefereed.
Yeah. Any other questions?
Did the Mount Sinai, this is from the actual work is the Mount Sinai group?
Yes, this is all Mount Sinai.
This is
it's Florian's assay. I don't think he led the study.
Yeah, plus Florian plus.
Right.
Did they describe any of the work that they have done in this paper with who among the healthcare workers was infected and who not?
So the only
epidemiology.
Mm-hmm. So they didn't really go into details about the epidemiology. The only time they mentioned the healthcare workers was in the first part of the study where they were screening for seroconversion. So they not only screened individuals whenever they could get them in, they started in March. So the current individual count was over 51K. That was as of July 2nd. And they mentioned that in addition to, screening patients that come in, they also made it available to any healthcare workers in their system, but they didn't give direct statistics as to how many healthcare workers were screened.
Right. There may be another paper because I've heard Florian report or the Florian's co-workers report that the data from the healthcare workers suggests that a lot of them acquired infection from the community.
Mm-hmm. So another thing that they do mention is that when they're talking about the first figure with regards to spike antibody titers in the individuals, they say that because the screening program shifted from plasma donors, which were the patients, to employee screening with probably a higher number of asymptomatic infections, that they just mentioned that because to provide explanation for the decrease in fluctuation in one of their figures, which could indirectly hint at infection because of exposure in the healthcare system. But that's pretty much it in this paper.
Yep. Do they also, at this point, they are not reporting a two-stage test, ELISA test, are they?
No.
Because at first they were doing a receptor binding domain and then a spike. So those that were positive to the first.
Yes. But I believe the only actual time that they used the ELISA test was in the first part of the study. And then from those, from what I gather, is that they used smaller subsets of plasma donors that were already screened with ELISA for the second and third parts of the study. And that's to my knowledge. It was a little unclear, but I tried to read and look as much as I can, and that's the conclusion I come to. Okay, and then I'll hand it off to the last person who's doing the summary of the new article.
Thanks, Amanda. Kat, why don't you?
Yeah, everybody's all, when I said they were all at Twitter, I didn't realize
That it was actually on Twitter. I know. I thought there was
actually a Twitter.
it was a good pun without even meaning to.
I do talk about it a little bit. Ryan, if you want to make me a co-host, I can start my slides.
I think anybody can share, but.
Oh, wait, I think I can.
Yeah.
I'm good. I use Zoom every day now because of the classes I'm taking, and I just forget how to use it. All right, so we have 9 minutes left, so I will do this quick and dirty like most COVID science that's coming out lately.
And so there's not a whole lot in the paper to do anyway. I mean, it's
exactly, yeah.
And then we'll destroy it.
Yeah. So this is a correspondence from a research group to the editor of New England Journal of Medicine, and it was the rapid decay of anti-SARS-CoV-2 antibodies in persons with mild COVID-19 illness. I picked these kind of classic sci-fi horror movie posters because that's how a lot of headlines feel like these days. So to put it mildly, letters are the new New England Journal of Medicine first authored papers. So if you're looking to get published, just write them a letter and it'll make national headlines.
Well, actually, that's not how it happens. I got involved in one as a referee. Because they send papers in which are just observations. And the editors, if they like it, tell them write us a letter. Format it as a letter.
That's interesting.
Yeah,
well,
that's how it started at first. It may be more of a routine now.
Okay. So just very briefly, I won't get into all the immunology behind plasmablasts and B cells developing antibodies, but antibody decay is the rate at which detectable antibodies diminish post-infection. Which antibody you measure matters. So IgM is a large pentameric antibody. It's produced earliest in response to infections, and IgG is the most abundant antibody found in humans. And this may take a while to form post-infection or immunization. So IgM, you're sick now. IgG, you have developed a threshold of immunity. Anti-receptor binding domain antibody levels is what we are measuring with the assay, whether it's ELISA or hemagglutinin inhibition assay for influenza, for example, and that corresponds pretty well to plasma viral neutralization activity. Long et al., which this correspondence was a response to, reported a rapid decay of SARS-CoV-2 IgG antibodies early in infection, but this letter pointed out that they didn't really give a lot of details as to how they found that conclusion. So this was kind of here's what we did to validate it. So this researcher, I hope I don't say this wrong, Ibarrando et al., they had 34 participant blood samples that were analyzed. 88% of them had a confirmed SARS-CoV-2 PCR assay, and only 12%, which was 4 people, had COVID-19 symptoms and reported living with or cohabitating with persons known to have COVID-19. And I really don't like this because those people were not tested due to having a mild illness. So it's only a small percentage of our sample, but I think it's important to note because we already have a really small sample size to start. So 31 people in the study had 2 serial IgG antibody measurements and 3 had 3 serial measurements. It obviously would be a lot better if they all had 3. I'm I'm sure it's just difficult to get people to come in, but I think that was another limitation to point out. And then another really big issue was the range around in which they measured serial measurements. So it was an average of 37 days after the onset of symptoms, but anywhere between 18 and 65 days, which is crossing the range into the last measurement range. If you can see that, they're kind of like right on top of each other. I understand that it's difficult to get people to come in and have samples drawn. It's really time consuming, but it's got to be better than this. And their observation period was 90 days. So I don't understand how they were including people beyond. It just seemed very like sloppy and quick. And then if you heard Arnold when we first all got on, they use linear regression, but antibody decay isn't linear, it's actually exponential. So they did a linear regression adjusting for participant age and sex and the day from symptom onset to the first measurement and the log 10 antibody level. They reported that there was an initial mean IgG level of 3.48 log 10 nanograms per milliliter. That doesn't really translate into much. So they translated it into the decay from their linear regression. The slope was negative 0.0083 log 10 nanograms per milliliter, translates to approximately a half-life of 36 days over their study's 90-day period, the time that it took for the antibodies to decay. So here is their figure from that paper. I think it's a nice spaghetti plot of the log transformed IgG concentrations plotted against the time since the onset of symptoms in each participant. So this does look nice because you can see, you can trace pretty well individual changes in antibody titer, but it's, does anyone want to comment whether they think this looks rapid? You don't have to, that's kind of like a rhetorical question. But it's sort of this idea that like grabby headlines like that are what get published. So to me, this doesn't look very rapid, and to a lot of other people, it doesn't look very rapid.
Well, not only that, things are, we know that the most rapid period is right after infection or vaccination, and then it levels off.
Yeah, I think that's the one thing that's missing from all of these studies is we don't know what the correlative protection is, right? Like
And we don't know, not only
what antibody level is protected.
Protection. There is no standard. The FDA, in conjunction with the British equivalent, is trying to develop a standard serum.
Yeah, and I think we also know that antibodies, that people with higher antibodies titers wane faster, right? So
right.
This is something we know from Josh's work in flu.
Yeah, that's why I emailed Josh, and he was unable to come on to the meeting today. But we know it. Josh has documented this for flu. It lingers on for a long period afterwards, after an initial big drop.
Yeah, definitely. So this correspondence at half paper's conclusions early antibody decay after acute viral antigenic exposure. This was approximately exponential. The change in titer was statistically significant and quicker than that reported for SARS-CoV-1. These findings were consistent with Long et al., and humoral immunity against SARS-CoV-2 may not be long-lasting in those who experience mild illness. So then they very, very briefly, in like 2 sentences, went on to discuss some limitations. Basically, this by no means means that there's an indication for immunity passports or herd immunity, and this might have implications for vaccine development and vaccine durability. But also, I don't think there's many conclusions from this. So some more important questions to kind of look at is, what is the long-term baseline titer? How long lived will that titer be? How much antibody is necessary to be protected against a reinfection? And how much antibody do you need to be protected from disease? Those are things that are kind of still floating around that we don't understand about this disease. So in my real conclusion, do better science, you'll get more realistic headlines, and you can improve science communication and trust in our field. And don't be toxic on Twitter, or just trust something because New England Journal of Medicine or Lancet, Nature, Science published it. I was a little surprised that someone would talk to Florian Kramer like this, but I guess no one cares when you have the screen in front of you. Any questions?
I actually have a really quick question, and forgive me if I phrase it poorly. I'm just a measly biology student. So I saw that the findings were consistent with Long et al. And the paper that I also presented talked about Long et al. And how the target antibodies were mainly nuclear protein antibodies. So was that also the same in this paper / headline grabby thing?
These are a spike, I think.
Yeah, they used the spike in this one that Florian used, I believe.
Okay.
Yeah.
That was just my question. Thank you.
Thanks.
Thanks, Kat.
Thanks, y'all.
Don't be toxic on Twitter. That's good advice for everybody.
I could not believe that someone was talking to Florian like that. I wanted to say, like, do you know who you're talking to?
So for
Right, I don't
those,
know how to get out of here. Oh, there we go.
so for those unfamiliar, Florian Kramer is probably the foremost expert on, and certainly SARS-CoV-2 antibodies, but also influenza immunology. He's a very highly regarded expert.
And he
And
likes
certainly-
neuraminidase too.
He certainly knows how to read a plot, a spaghetti plot. Okay, so thanks everybody. And I will take suggestions over the weekend for topics and send out a new topic for next week, hopefully Monday or Tuesday.
I have a quick question.
Sure, Iman.
I'm signing off because I got another one to do, Ryan.
So tweets like those tweets go away fast because over time they vanish. But such papers remain on Google Scholar and other repositories. So what happens? Are these papers going to be cited again and again without being criticized?
Well, there's a lot of retraction. I mean, there's been quite a few retracted papers already during this pandemic, but I think the other thing is that they'll be cited, but they'll be cited often as, you know, examples of what not to do, or of a sort of a weak analysis that could be strengthened in a new way with a new method, new analysis. So,
but if I've not read those tweets and I'm looking for literature about the topic, there's no reason for me to figure out the issues with that study. And I may cite it in my study.
it's true. Yep. And then it would be up to whoever reviewed your paper to say, you cited a method that has not previously been well received, or has known flaws, and you should rethink your method. And I mean, that's the way that it should work. Now, whether it does work that way or not is, you know, an open question, but that's the way it should work.
Right.
All right. Well, everybody have a good weekend. And then, like I said, if you have an idea for a topic for next week, please feel free to let me know. And we'll see you next week.
Bye. Thank you.
Thanks, everyone.
Ryan. Bye.
Thank you, everyone.
Thank you.